Welcome to Coffee and Conversation. My name is Honorine, and today I'm with Courtney Whitcher, who is pursuing a PhD in ecology and evolution. Courtney, thank you for being with me today.
Of course. Thank you so much for having me.
Let's start with a little introduction.
Yeah, so as you mentioned, I am Courtney Whitcher. I am a PhD candidate. I'm just finished my third year in the Dr. Emily Moriarty Lemon Lab here at Florida State University. I am originally from southeastern Michigan. I went to college and got my undergraduate degree at the University of Michigan. That's where I kind of got into research, but I'll get into that in a little bit. And then I really enjoyed everything I did there. I worked in the museum, and now I'm currently here. I loved Florida State as a place to, the amphibian diversity is incredible here. And so that's something that brought me here. And then other stuff about me besides research, I love being outside. Tallahassee is great, all of the natural areas. I also am involved in the community bands of Tallahassee. So in the concert band and the jazz band, I do a lot of knitting and reading. And yeah, those are some of my hobbies as well.
At the University of Michigan, did you do your BA and MA, or did you do your BA and then you went straight for a PhD at FSU?
Yeah, I just did my bachelor's of science there, and then I had about half of a year off where I worked as a teaching assistant and did research, and then I came here for my PhD.
Oh, awesome. So you were a teaching assistant with basically just a BS at the University of Michigan?
Yeah, I got to teach and be involved in the first herpetology or biology of amphibians and reptiles class at the University of Michigan with an all-female teaching team for the first time. And so that was a really great experience, and I learned a lot, and also added to my love of amphibians in my own research.
That's amazing. Was it your motivation to pursue a graduate degree?
Partly. So a lot of my experiences at the University of Michigan helped that. So I kind of had this nice additive story of I got to the University of Michigan. My first research experience was looking at enhancing biomolecular fluorescence using metal nanostructures, but then I was missing the outside field aspect and working with actual animals. So then I got a job at the University of Michigan Museum of Zoology, where I worked for a couple of years in the herpetology department, or biology of amphibians and reptiles, in that department of the museum, where we have 15 million specimens. And then that led to a project of me looking at tropical tree frog communities. And then in my final year, while I was looking at grad schools, I knew I loved research and wanted to learn for a career. The first biofluorescent frog was discovered. And so my two worlds perfectly combined, where I had been studying fluorescence and then studying frogs, and I loved them both. And then they came together in this discovery. And then I came here to pursue that for my dissertation and try to figure out what frogs biofluoresce and why and how. And so it just kind of nicely added together for me. A lot of luck happened there.
So you study fluorescent frogs. Tell me more about that. What do you just said, you study why, how, but I don't know anything about fluorescent frogs. So tell me more.
Of course. So my dissertation is uncovering the evolution of biofluorescence in tree frogs specifically, but I'm looking at the function, the mechanism, and how that might affect speciation in the group. So like I said, the first biofluorescent frog was discovered in 2017. So it's a pretty new discovery. We don't know why. So pretty much, you guys are probably familiar with bioluminescence. That's how fireflies or the lure of an anglerfish glows. But biofluorescence is a little different. It's another way that species glow, but they take light from their environment and then they re-emit it at a longer wavelength. And so it changes the color that they can see. And so my work is looking at first just trying to figure out which frogs have this feature and what colors they emit that light at, what colors they glow in what areas of their bodies. So that was part of my I just went on an expedition to South America to document the diversity of biofluorescence in frogs. And then I also do mate choice experiments and predation experiments to try to see what how this biofluorescence may be affecting the behavior and evolution of the species. And then I'm also, on my expedition, I also took skin samples and genetic data and trying to understand both the genetic and the chemical mechanism. So we know that this is a trait that some frogs have, but we don't know why or how in most species, or even if most species have it. So I kind of have an observational part of my dissertation and then trying to dig deeper into those questions for a couple species.
I guess I'm going to ask the tough question that no one like to answer, but I will ask. Why study fluorescent frog? Like the so what question?
Yeah, there are a lot of really interesting reasons. So first, it's important to just understand, in order to protect species, it's important to understand what features affect how they reproduce. If fluorescence is important for mate choice or predation, or these life history traits, these traits that affect how a frog survives, then if we can understand those better, we can better protect these species. But also, so that helps with conservation aspects, but then also fluorescence has a really cool added implication where you may have heard of green fluorescent protein, or GFP as it's called. That is a fluorescent protein that was sequestered from jellyfish, and in 2004, I believe, it won the Nobel Prize for its advancements in medical applications. So if my work looking for the mechanism of biofluorescence in frogs also happens to find new fluorescent proteins that fluoresce in different colors or at different ranges, that can really open up its uses in medical applications. So if we can to track images at and processes at a very small molecular level. So we use green fluorescent protein to attach to molecules or cells that we are interested in looking at. And because it's a natural biological product and because it doesn't interfere with anything else, we're able to use that to track the function and the movement of some of these processes. And so if I can find new colors of fluorescent proteins to use, then you can tag multiple things and look at them all at the same time. And so it has a lot of potential to also help in the medical field, which is a really cool extra aspect.
Wow, that's awesome. And so you mentioned your expedition. So you recently came back from the Amazon,
yes,
where you were doing field research. Tell me more about this expedition. Like, and also maybe how did you finance it?
Yeah, so I got a few grants to finance it, some from the Society for Systematic Biologists was the big one, and then a couple of smaller ones from Florida State University or local herpetological societies. And like I said, the goal of this expedition was to go around, I went around South America to try to document the presence and diversity of biofluorescence in frogs. So, and in amphibians in general. But since the past few years, about 35 species have been tested for biofluorescence. About half of those were tested by myself or my collaborators. And we were finding that people had tested frogs for biofluorescence, and in some they found it. But we had tested some of those same frogs under different excitation wavelengths. So if you remember, fluorescence is taking in light and then re-emitting it at a longer wavelength. So depending on the wavelength they take in, you may or may not see that fluorescence. So you test by shining a specific wavelength on the frog and then measuring the emitted light. And so we were seeing that researchers had tested frogs and said they weren't fluorescent, but then we tested them at a different wavelength and they were. So what I did is I took down a huge range of wavelengths, like a 200 nanometer range. So I had 5 different excitation sources. I wanted to specifically focus on tree frogs because that's my dissertation, but I tested a lot of other things too. So we went to 8 sites across South America to try to cover that diversity of tree frogs and their range and where the different habitats they live in. So We had a site in Ecuador, in Colombia, in Peru, and then 5 sites in Brazil. We had a few in the Eastern Highlands, 2 in the Amazon basin. And at each site, I collected emission spectra, so the fluorescence emission under each of those light sources. I got photographs to calculate biofluorescent patch size of each frog. I took samples of biofluorescent and non-biofluorescent skin to look at gene expression between them, as well as try to see if I could get that fluorophore, that fluorescent chemical I was talking about, that mechanism sequestered out. I took genetic data for phylogenetic analyses, and then I accessioned all of my specimen at the local museums in those countries. And so we ended up getting, we tested 528 individual amphibians from 165 species, 41 genera, 16 families, 3 orders. So we were able to test, there are 3 groups of amphibians, frogs, salamanders, and caecilians. So we were able to test across all of those. About 60% of my individuals were hylidae or tree frogs, so from the tree frog family. And so I was able to get a lot of data. Like I said, no one has ever tested these frogs for biofluorescence. So anything that I found on this trip is new. And now it's just trying to go through all of that data and figure out which species could be using this in different ways. And how this biofluorescence may have evolved across the whole amphibian and tree frog tree. So why we see this trait and how it functions.
What a trip! How long did you stay in South America?
Yeah, so it was 10 weeks total. So we had about a week-ish at each site.
It must have been exhausting.
It definitely was. It was exhausting, but a very good and successful trip. And I got a lot of data for my dissertation. And so, and also made a lot of like great connections with collaborators in South America, and learned a lot about culture, and had all these extra things that I also got to gain besides just the data while I was there as well.
So in your grad impact profile, you said, I quote, "I'm proud that I have worked hard to maintain my hobbies outside of my research career." And then when you introduce yourself, you mentioned, loving being outside, you're part of a community band, you love reading. So how do you manage, it seems you're doing so much. How do you manage life outside of grad school?
Yeah, so I think part of it is just making sure that I continue to do those hobbies and make time for them. So that means that I try to, on Mondays and Wednesdays when I have band, and then I finish before I have band, and I get to go do that in the evenings, and like keeping that time outside of work or after work hours, quote unquote, where I am still doing things that I enjoy. I love my research. I absolutely am so interested in it, and it gives me so much joy. But I also have a lot of other interests outside of research, like band and knitting and being outside and reading and all of these things. And so I think it's just I prioritize that just as much. Sometimes I prioritize each of these things that bring me joy, and try not to. I think in the science world, it can be very difficult to not devote your entire life and every waking hour to your research, because there's always more, and the more you find out, the more questions you have, and I think it's important to also realize that doing the things that you love and that bring you joy outside of that only give you more energy and like make you a better person to be able to then address the scientific problems and science side of work. And so I think it's just all about trying to find that balance and working with it as you go. And everyone has that looks different for everyone, that balance of work and life. But yeah, I don't know. I think that once I got into grad school and realized how enveloping research can be, that I also decided to prioritize doing things that I enjoy and keeping those hobbies up. So
my last question is, how do you see yourself in 5 years from today?
Yeah, that's a great question. So Hopefully, so I'm going into the 4th year of my PhD. Hopefully in 5 years, I want to go into the world of academia and be research faculty at a college or university. So hopefully by that time, I'll either be finishing up a postdoc and starting a position as some kind of research faculty at a college or university, and continuing to do research that I love, whether that be, I enjoy biofluorescence, hopefully it's still in that, but also a lot changes over 5 years. You always find new questions that are exciting to you. And so I'm guessing still something in trait evolution to some effect, but yeah, I see myself getting to that more final stage of, not final stage of my career, but the point where I am more established and starting to maybe develop my own lab and see research from a different aspect, and hopefully teach like a field class as well. And so that's my goal for 5 years, is to be in that position, or at least still working towards becoming research faculty at a college or university.
Well, thank you so much for your time. In your busy schedule, I really appreciate it. It was great to get to know more about you and your journey and your expedition. It was very interesting to learn more about frogs.
Thank you so much for having me, and I had a great time talking to you.